Hello, ladies and gentlemen. Today we are going to talk about an interesting topic, that is about how the fire is controlled in our bodies. That is how the bioenergetic control works through mitochondria and the respiratory system. So, let me share my screen. Yeah, so the title of today's talk is Bioenergetics Taming the Fire. Now, this is the fifth talk in the series of 12 talks related to biointelligence. Uh, this is a picture that I happen to have liked on internet. It's an artistic representation of mitochondria, which is said to be the powerhouse energy control center within the cell. Yeah, so it's an artistic representation just for visualization. Okay, and I'm thankful to somebody who has put this one on internet. It's a wonderful thing. So due acknowledgements and due respect. So in this video, like all the videos in this series, all the pictures are from public domain, and whatever is there in yellow highlight is my own thoughts. Okay, so uh, let us first see what are the examples of energy requirements at system level or physiological level on the body as a whole. Of course, the most glamorous part is to power the ionic currents in the nervous system, okay, related to cerebral intelligence and nervous intelligence. Then, of course, to carry out the movement of food, movement of blood, circulatory system, the respiration, and muscular contraction and relaxation to power up these movements, and for applying gentle squeeze to the glands to take out the juices, to constrict the vessels or vesicles, to control the flow of fluids in the body, etc. And maintaining the temperature, pressure, chemical level, and various physiological parameters in the body. Some energy is required towards that. And of course, to control the anabolism and catabolism in terms of chemistry. Okay. So to power the chemical reactions, etc. Yeah. And coming to the cellular level, intracellular level, the energy requirements are there for opening and closing of channels and walls in the membrane, including ionic channels for neurons. Yeah. Movement of vacuoles or organelles and change of cell shape. Okay. By some contractile movement with micro muscle within the cytoplasm. And then active membrane transport. Yeah. Active pumping through the membrane, etc. Then extrusion of cell produced fibers through membrane, whatever secretions, excretions are there through the cell membrane. Yeah. Then propulsion of molecules like mRNAs, activation of rRNAs, movement of tRNAs, and so on, as far as genetic translation is concerned. Then also talking of genetics, energy is required for zipping, unzipping the DNA, for carrying out the transcription and translation of the genetic material, and intracellular anabolism and catabolism, that is protein synthesis and working with energy molecules, etc. Yeah. Then to carry out the cell division dynamics, mitosis and meiotic cell division, to cause the cell death if need be. And at molecular level, of course, to do twisting, turning, splicing, shaking, vibrating, breaking long chain molecules into simpler ones. Okay. So all these are by far the energy requirements within the intracellular processes. Now, the question is, uh, okay, we are taking oxygen through lungs and we are having the fuel derived from the food that we take. Okay. Now, there is fuel in the blood and there is oxygen in the blood. Okay. So, what is there to prevent a fire happening within the lungs itself, within the blood itself during transit? So, there has to be some passivation of oxygen or fuel or both to prevent a fire from happening where it is not required. So, that's why the title Taming the Fire in biological systems. So, oxygen is absorbed in lungs and carried everywhere through the blood circulation. So, also food molecules. Ultimately, they come through digestion and are circulated through blood through bloodstream all over the body. So both oxygen and fuel are there in the body. They can mingle, they can burn right there. Okay. So what prevents the fuel to burn right within the lung itself or while it is carried, while it is in transit? Okay. So obviously, like we bottle up the fuels for safety, we have to bottle up the oxygen, okay, while transporting until it reaches within the cell. Ultimately, whether it is physiological level of energy or ultimately everything happens at intracellular level. Okay, so until it reaches some cell and the cell is able to take the control of that energetics, it has to be passivated by bottling it up. So, how is the oxygen bottled up while transporting with the blood? And also within cytoplasm, because as soon as it enters the cell, it should not start burning. It should burn where the energy is required precisely. Okay. So it has to remain passivated until the site where the energy is specifically required. Right. From engineering point of view, that is the call. Okay. So how it is bottled up, how it is released, how it is reached where energy is required, how it gets controlled, because if it is fire, it's like our stove, you have to have some amount of flame control. Some controls have to be there. So like what are the controls which prevent? I mean, otherwise if it just depends on availability and intake, then it will be uncontrolled fire. Now in biological system within the cell, you can't have uncontrolled fire, obviously. Okay. So how is the fire controlled within the cell? That's the main interesting talk. Now the bottle which carries oxygen is of course hemoglobin. Okay. So hemoglobin is a complex molecule, as you can see in the picture for visualization. I'll not spend too much time because this is some from public domain, some information just being given for your reference kind of a thing. So oxygen is absorbed by hemoglobin. So you can see some oxygens getting attached here. And once it is taken up by the cell, it gets stripped off. And sometimes some CO2 gets clinging to some of these, but that's a small percentage, of course. So basically, these are the bottles that carry oxygen safely in the blood until the cells. Okay. So oxyhemoglobin, which gets deoxidized, so deoxyhemoglobin returns. Okay. So that is the uh, molecular bottle which safely supposed to carry the oxygens. Okay. So both fuel and oxygen are possibly transported and carried around in passivated state. So they are only deactivated through respective carrier molecular appendages and they prevent burning during the transit. Okay. And these appendages, of course, the molecules that carry the so-called bottles have some other properties also, that they have to be identified by the cell surface as a valid entity which is supposed to transfer things to the cell. So cell has to acknowledge and open the gates, right? So the shape of these molecules is identified by the proteins that reside on the cell membrane surface, and so that it is programmed to open the gates when this arrives, and it's supposed to take in the oxygen from these ones. Okay. And of course, similarly, fuel will be taken from the molecules that bring in the food part. Okay. So they are selectively ingested within the cell. Not anything and everything gets ingested by the cell membrane. Of course, it is supposed to be intelligent to identify what it is supposed to take in and what it is supposed to disallow. Okay. So after oxygen has been stripped off the hemoglobin bottle, it returns and it is supposed to carry the CO2. Okay. So CO2 gets converted into some acid, carbonic acid, which is supposed to maintain the pH of the blood, by the way. And that mechanism is about 85%. Some CO2 can also get directly dissolved in plasma because CO2 is relatively harmless. Oxygen can cause fire, but CO2 cannot cause fire with something. Okay. So CO2 can come in dissolved state, it can come in whatever way it can. Uh, and it can get released in the lungs safely without any problem. And but most of it gets chemically absorbed as carbonic acid, and which is transformed, and the CO2 gets released in the lungs. So oxygen, when oxygen gets absorbed, CO2 is given up. Okay, so that is the transaction happening in the lungs. So oxygen is taken in into the hemoglobin, and CO2 from this reaction is liberated, and also whatever is absorbed on top of the hemoglobin also goes up. Okay, due to pressure differential. So ultimately CO2 gets released. That's how the loop gets complete. The oxygen is taken in, CO2 is liberated out back to the lungs. Okay. Now the carrier within cytoplasm, similarly, the bottle is called as cytochrome. Cytochrome is carrying the oxygen. Okay, so it binds with the oxygen. It has little bit of iron part from M. Okay, it has some copper part, and it has some. It's a complex. It's not a single molecule. It's an assembly, as you can see. Okay, so oxidized cytochrome is supposed to be the bottle that carries oxygen within the cytoplasm. Okay, and being relatively smaller molecular assembly, this can freely diffuse into the cytosol, and it can carry the oxygen everywhere in the cytoplasm. Okay. And then after the oxygen gets removed from this one and gets used up, the deoxidized container is returned by the way of diffusion back to the cell membrane, and from there it can pass the CO2 to the process that is going in a similar way as it is happening in the lungs. The byproduct of chemical reaction, that is CO2, is taken up wherever the oxygen is absorbed, and it goes back. So that's how the cycle gets completed. So the problem is, it is supposed to deliver energy in measured quantity, and cytochrome gets stripped of oxygen, and burning of the fuel and molecule gets permitted in a controlled manner, and it happens in an organelle which is called as mitochondria. So mitochondria is an autonomous entity within the cell. So cell is not something that is controlling everything centrally, or that has been designed to evolve centrally with the central, structural database kind of a thing. So mitochondria is actually, it is coming from a simpler prokaryote cell, and somehow during the process of evolution, those simpler cells have got embedded into more complex cells. Okay, eukaryote cells. And so mitochondria is actually evolved from simpler cellular organisms, which have got planted as a parasite in some other cells. So a cell is not a single entity. It has life within life, you can say. It has living mitochondria within a living cell. You can put it like that. So mitochondria, like bacteria, can replicate autonomously within cytoplasm, and their population density and their disposition and orientation gets aligned according to the energy requirement within the cell. That's how the system has evolved. Okay. For example, they lie parallel to the rivers of ionic currents in the neuron. So ionic channels which flow like rivers within the neuron body, from dendrites to axon to the main body, etc. So wherever there is a flow of ions in a bigger way, parallel to that river, you see the islands of mitochondria lining up on either side. Okay. So it is supposed to be autonomous. It grows on its own. It senses the requirement and it reaches there. Okay. There is no central control which dictates that. Okay. So as I said, mitochondria have been believed to be have evolved from simple prokaryote cells. They have grown inside eukaryote cells. They carry their own DNA, a simple loop of DNA. They carry their own ribosomes, and they synthesize their own proteins also partly. Of course, partly they can borrow the proteins from the cell, and they also borrow some ribosomes from the cell. They could, and they have got some specialized role of an autonomous powerhouse and control center for the energetics of the cell. Okay, so and cells like muscles and neurons, which are very intense in energy, which have very specific localized requirement of energy, they have a larger population of mitochondria, and they reach wherever the energy is required by themselves. So this is, of course, how the mitochondria looks like. Uh, Approximately, these are the dimensions. The length is about 3 to 5 micron, width is about 0.5 to 1 micron. Outer area is smaller than inner area. So there is an outer membrane and there is an inner membrane. The surface area of inner membrane is bigger. So it has to fold in like shells called cristae. Okay. And in the interspace, which is there there is supposed to be a matrix. It is containing a fluid which is isolated from the main cytoplasm. So it is a local island of a specialized fluid that is there within this one. And within the matrix also there are loops of DNA which are supposed to synthesize some proteins that get planted into these outer and inner cell membranes. Okay. So double membrane shells are there. Inner is bigger than outer, and inner matrix contains isolated fluid having ionic solutions for the chemical interactions within the matrix. And outer wall contains some protein globules to control the transactions with the cytoplasm. Okay, so they interface with cytoskeleton, cytosol, and allow or disallow certain things to travel across this membrane and go in the interspace. So this is how the mitochondria. So clearly, this looks like a very simple bacteria which grows within the cell. Okay, this may not have proper nucleus since it is prokaryote cell. It has just a loose DNA. Okay, and it has rRNA. So all the rudimentary processes of a cell are there. Only elaborate nucleus is not there. Of course, this is mitochondria itself, so this can't have another mitochondria inside. So So this is the simple thing. It works like powerhouse of the cell. It takes the oxidized cytochrome from cytosol, which we said is the bottle that is bringing in oxygen within the cell, and that oxygen is removed in a controlled mode for different processes, 5 different processes that are there inside, and the deoxidized cytochrome gets released back to the cytosol for reuse and recycling. Okay. And mitochondria, basically what it does is there are some energy molecules. Uh, the main energy from the cell comes from adenosine diphosphate uh, getting converted from ATP, that is adenosine triphosphate. So ATP breaks into ADP and it releases energy. And inside, of course, there is a big elaborate chemical cycle of oxidation reduction reaction called Krebs citric acid cycle, which we shall see for reference. Though I have no intention of going in the chemistry details of the whole thing, that we leave to the biochemists. So I'll focus more on the engineering aspect of the control part and the intelligence part rather than the molecule part and specific molecule part. Not going into specifics, but just for the namesake, yes, when ATP breaks into ADP, it releases energy, and it can further break into AMP, it is adenosine monophosphate, and there is a cyclic version of it where the molecule gets connected back like a ring, so it's called cyclic AMP. Okay, so and ATP, having got energized in mitochondria, is allowed to diffuse through the cytosol, and it reaches everywhere, and it provides energy at the molecular level to various enzymes and ribosomes, and uh, wherever it delivers energy it degenerates to ADP and AMP and cyclic AMP, which diffuse back again to mitochondria again to get converted to ATP. Okay, so that's the main, this is the main media which is carrying the energy to the site. Okay, the fire has burned, the oxygen has been taken away, but the fire burns where the ATP gets converted to ADP. Ultimately energy is provided there. Okay, so energy is specifically reached in controlled manner to where it is required by the amount what is required by the processes. So there is no surplus energy, there is only enough energy to do what they are supposed to do. Okay, good. So It is highly optimized, energy optimized system. So external membrane from mitochondria is supposed to pump the ADP inside and AMP inside and cyclic AMP inside, and it's supposed to give back ATP to the cytoplasm. And also it is supposed to pump in the oxycytochrome and supposed to pump out deoxycytochrome back into things. So that completes the total transactions related to energetics, right? So these are the summary of various names that you will see in some of the pictures. As I said, I have no intention of going in the details of chemistry, but for those who are interested, of course, the molecular structure has been given for adenine triphosphate. And so this is adenosine. So this is adenine and attached with ribose. So that's adenosine, and this is phosphate. So triphosphate has three such chains. Diphosphate has only two. One bond gets broken, and this high energy bond releases energy. So when ATP degenerates to ADP, it releases about 30.7 kilojoule per mole. So that's the specifics for somebody who might be interested. There are all these energy molecules are basically derived from nucleotides. Nucleotides are the substances that adenine, thymine, which form the DNA. Okay, so adenosine and thymine are nucleotides. So also xanthine, guanine. Okay, we remember. So basically, their phosphates are the energy molecule, energy carriers which carry supposed to carry the energy everywhere. Okay, so sometimes there is a single, sometimes there is a di enzyme like NAD, nicotamine, adenine dinucleate. So there are two nucleides tied with a nicotine amine molecule. Okay, similarly, flavin FAD is having two adenines, right? So basically, these are all adenine, guanine, xanthine based molecules. Okay, which and all of them are used in in mitochondria, but primarily we are focusing on the most important one, that is ADP to ATP transition. So we are not discussing all others which are there, but they are also there. So just remember that in a similar way they will also be there in a smaller way, but they will be also there, right? So the main thing is to illustrate the function, transition from ADP to ATP, and of course energy being liberated by ATP wherever it is required within the cytoplasm. So this is the vicious circle. This is the Krebs cycle, which is a complex chain of reactions. Here you can see that ATP ADP transaction is there, NAD NADH transaction is there, okay, FAD FADH transaction is there. So all these are basically oxidation reduction kind of transactions with the electron chain, and so it's like a multi-stage cycle with several spokes, with several cascaded processes. I'm not going into chemical details of what are these things, okay, but it is multi-stage cyclic process with several cascaded parallel processes which are required for different processes. Okay, so everything is useful here. Okay, so different processes are there which are cascaded together in a bigger wheel. Okay, so they are running parallelly within mitochondria, and any of the process can throttle at every stage. It is possible to throttle. It's possible to control the speed of the loop. Okay, so basically the slowest process will decide the overall cycle rate, right? So overall pace is controlled by the slowest process, and these are the participants which are there. Participants, and CO2 is the byproduct, as we said, which is supposed to go back. So that gets liberated and it gets carried by the deoxycytochrome. Okay, and basically this cycle ensures that multiple factors which are there to control various processes of the complex cell system, they all get coordinated and they are kind of synchronized, okay, on the whole. So there are many parts in this, and many of these things do many processes, but the whole thing gets controlled by this bigger cycle, and they are of course interdependent. So level of activity, it depends on the slowest process or the input for the slowest process. So controls are there, The rate of energetics is controlled through this one, through this big wheel that is there. Okay. So in physics, normally we say that energy like heat is directionless scalar quantity. But you will appreciate that in engineering, the activation, for example, if you have a hydraulic cylinder or a solenoid valve, it releases energy in a directional way, specifically where it is required to do the actuation, right? Similarly, at molecular level, actually you require a vector, a force vector or a momentum vector, because to carry out smaller changes in macromolecules, okay, you need to hit it with something like a carom board striker. Okay, so you have to provide a momentum vector precisely of the measured magnitude in a measured direction at a measured place in the molecule to bring in the transaction most effectively. Otherwise, if you depend on heat, you just jiggle the whole thing, increase the average kinetic energy of everything that is vacillating, and then you hope that after 100 attempts something will hit the right place and the process will happen. Now, obviously, such wasteful processes are against the optimization principle. And as an engineer, I hope and believe that in biology things are much more optimized than that. So hit and trial and multiple trials and all, it's not likely to be there. Everything is aligned, everything is designed to work for shot. Okay. Okay. So you have to have a mechanism to convert the energy into a momentum or a force vector, specifically where it is required. So this is a big ask, that okay, burn fuel in oxygen within mitochondria is one thing, but how do you take that energy and convert it into the vector where it is specifically required? That is the intelligence part of it, and that's the engineering part of it. Okay. So there are of course transferases enzyme in biology. They assign everything to enzyme and append word ASE in that and represent the same function. So transferases are basically enzymes which are wonderful naturally created mechanisms for controlling the passivated energy, energy where transport, release of measured electrical, mechanical, or inertial momentum at precise location and orientation at molecular level to specifically twist, index, tension, splice, or move the molecule, vacillate, or oscillate the molecule in a predictable manner. Okay. So all this control from scalar to vector is supposed to happen by these kind of transferase enzymes. Okay. Where from a general heat-like thing, it is transferred into a momentum vector. So energies in the form of heat will generally promote wasteful random jiggling, as we said, with some statistical probability that sometime it will hit the right thing, and so the process will progress. Okay, but it's unlikely that biological systems will be that inefficient. So talking of the mitochondria, of course, the outer membrane has several functions. It has got some proteins embedded, like all the cell membranes have. So it controls the flow of cytochrome both ways, that is from cytoplasm to inside and inside mitochondria to cytoplasm. And it is having proteins that are attached and anchored to the cytoskeleton. So in a way, the mitochondria body itself locates itself with respect to cytoskeleton. Okay. So, and it can even control the tension of cytoskeleton fibers. So, to put it lightly in another words, it can fly kites through the horizon of cytoplasm by string pulling, literally. So, cell, if you say it is like a tent, cytoskeleton is like a pole, and the ropes, the tension in the ropes is what will control the shape of the cell, shape of the tent. So, the tensioning arrangement is there in mitochondria. Otherwise, ropes will be there, tent will be there, membrane will be there, but it's a dead thing. It can't do anything. So, energetics control, force control is provided by mitochondria. So, it's a very important function within the cell. And of course, the outer membrane can control the ingress of proteins and ribosomes from cytoplasm into the mitochondria. And it can also control the flow of ATP, outward flow of ATP. Whatever ADP has been converted to ATP, uh, taking oxygen from cytochrome with that energy. And energy molecules which have been degenerated from ATP like ADP, AMP, CAMP, and some others like NAD, etc. They will flow inward from cytoplasm. Okay. Apart from that, mitochondria is having another function that it's supposed to store calcium. We'll come to that why it is supposed to store calcium. But calcium intake is also permitted by the outer membrane, and also fat and lipid. There is a storage of fat within mitochondria, and fat is supposed to be burned to create heat because some chemical reactions require certain thermostatic temperature control. So to achieve that temperature for to promote the chemical reaction, some of those fats get burnt, and lipids get used to build the membranes, the membranous structure of mitochondria itself. And of course, it can control water flow both ways. So the most glamorous part in the outer membrane that I was saying, that the cytoskeletal proteins, it's able to pull like this loop. Okay, pardon me, I am not exactly talking what is there in the figure. I am just talking what I want to talk from engineering point of view. So you see this opening, through this it is able to pull the ropes, able to pull the ropes of cytoskeleton. Okay, so it is able to form the loops, it is able to tension, it can control the tension of cytoskeleton, it can even pull the proteins fully bodily inside and use them to plant them as a valve within the membranes of mitochondria. That also happens. But then the more important function that I want to emphasize on is the control of tension of cytoskeleton fibers, which can cause locomotion of vacuoles, which can move things within cytoplasm, within the cell. They can also cause rupturing of lysozyme scission, thereby causing death of the cell. So there is also involvement of programmed cell death. Some of these strings, if pulled, they can rupture the lysozyme vesicle, and then it is corrosive enough to dissolve the complete cell. Okay. So under certain conditions, when it is deemed that cell is no longer able to fight the offenders and it's getting weak in energetics, as a last resort, it can commit suicide. So that is initiated by some of these. Okay. So mitochondrial proteins control, of course, energy metabolism. They control metabolism of amino acid, lipids, uh, this iron part, ham. Okay, and they can transport the metabolites. They can control the transport of metabolites. They can control the protein transport and folding of protein. Now, this is the folding of protein that is shown here. Then various signaling processes to exercise the controls, and changing the shape of membranes, etc., using the uh, lipids to form the membrane, these outer membrane, inner membrane itself. If mitochondria has to grow, if it has to multiply, because like a cell, it can cell divide. So it can divide. There is fission and there is a fusion in mitochondria. Okay. So two mitochondria can merge and combine into one, or one can split into multiple and grow, right? Like bacteria. So obviously it will require some hardware to build more membrane. So okay, that is there. Okay, and it can of course dissolve the proteins if required. Uh, it also builds in some proteins. Usually it draws some proteins from the cytoplasm and it modifies them with some of the internal proteins, and it combines both of them and plants them as walls, etc., within these membranes for internal use. So that also is supposed to happen. So proteins come, some proteins are directly directed to outer membrane, some are going in the middle interspace, some are going in the inner membrane. So that's what this is depicting approximately. So I'll not go into exact details of what this is, but okay, this is what I wanted to explain from this picture. So outer wall, proteins do the string pulling of cytoskeleton tension for carrying out the kinase, carrying out the movement within the cell, carrying out the locomotion of vesicles, vacuoles, particles, lysosomes to dissolve something wherever it is supposed to be dissolved in controlled manner. Okay, and it is not generally released at heat, but as specific force or momentum vector this way. That is, if you pull the specific string, you are directing the energy as a force vector, as a momentum vector in a controlled manner. So the main mechanism is cytoskeleton, which is controlling the direction of the force, channeling of the force. It can sort and direct. Of course, this is the proteins are ingested and directed wherever they are required internally. And it can also trigger the cell death by pulling some string that will rupture the lysozyme, as I was saying, and it results into death. Now, there is an outer membrane, there is an inner space, and there is uh, somewhere where I have given the values of this one. Yeah, see, this is the area of outer, this is the area of inner, this is the intermembrane gap, so about 8 nanometer. Okay, and the membrane thickness itself is about 6 to 7. So this is about thick as the membrane, slightly thicker than the membrane itself, the gap between the two membranes. Okay, so that's where we were. So coming to the inner membrane, there are 5 types of proteins or 5 types of processes, as I said earlier, that are getting controlled in the Krebs cycle, as we have seen. Of course, Krebs cycle, we have seen the Krebs cycle already. Yeah. So the there are 5 kind of proteins which are controlling this one, and those are here. Okay. So those are here. 1, 2, 3, 4, 5. Something which is controlling NADH to NAD, something which is controlling FADH2 to FAD. Then third is, this is basically the reduced cytochrome has been propelled out, and this is oxidizing cytochrome. So basically, oxidation of cytochrome is handled by this one. So, and this is converting from ADP to ATP. So these are the different proteins. So most of the proteins that do internal activities are on the inner membrane. Outer membrane things are what are interfacing with cytoplasm and what is selectively allowing or disallowing, or what is affecting the active transport across the membrane. Okay. The internal one is what is controlling the processes which are there in the matrix. So there are 3 cycles basically which are happening inside. One is conversion of cytochrome deoxidation and circulation, ADP to ATP conversion and circulation, and various transferases to move the ions and building blocks across, etc. So these are the 3 cycles which are controlled by these things internally. Okay, and this is just a slide of academic interest, entirely borrowed from public domain. So these are the proteins that are there on the inner membrane, and it is just saying that partly this mitochondrial DNA were 7, and the one which came from outside were 39. So together they were used to synthesize this protein. So the proteins are not entirely synthesized here; they are partly outside and partly inside. And the loop of DNA which is there inside, what you see these circles, these are carrying. These are shown in different colors, obviously. So 1, 2, 3, 4, 5. So you can match the colors. Okay. So this segment of DNA is controlling the synthesis of this kind of protein. So basically, this kind of correspondence is there. Okay. So coming to the inner matrix is supposed to carry out the Krebs cycle and activating control through the inner membrane, through the proteins that are there in the inner membrane. And DNA loop ribosomes are used for partial synthesis of proteins or enzymes to be planted in the inner membrane. and it initiates the division of mitochondria, merger, and lysing of mitochondria. Sometimes mitochondria gets damaged. It triggers its own lysing. So it invites lysosome to come and dissolve the mitochondria. And it can initiate even the complete death of program death of the cell. It can store fat. It can control burning of the fat to maintain the temperature within the matrix in order to provide proper environment for chemical actions that are happening in that fluid, which is isolated from cytoplasm. And it can use, of course, the byproducts, which are lipids, to grow the membranes further, to grow the size of mitochondria physically. And it can store the calcium. Calcium storage is generally used to control the contractile mechanisms which operate the valves on the membrane. So, like in engineering term, there are solenoid valves. You energize the valve, valve opens. So the contraction happens by using calcium because you require some kind of inorganic bigger force to do the actuations. So there the calcium ions get used. So calcium ions are used to have the inorganic brute force into the actuation mechanism so that it operates faster. It's not so gentle. And of course, solution form is there to store and diffuse ions and smaller molecules within the matrix. And it does have some sensing of level of ADP, and it's able to control the level of ADP. So it manipulates the valves in order to maintain the level of ADP and to maintain the, of course, the speed of Krebs cycle, to have the control on the total thing. Okay. So finally, mitochondria are seen to be adaptively present in larger number. In neurons, you see them lying parallel to the ionic rivers, and at the muscles also accordingly. And generally, they are seen to align with the cytoskeleton. So, with that, we end the talk today on this wonderful topic of cell energetics and how mitochondria does the control. So, I hope you have liked the talk today. If you have liked, please share, and please stand by for our next talk, which will be on neural intelligence. So, we'll talk about how the neurons work and how the human brain works, and how this wonderful aspect of biological intelligence functions. After having covered the basics at the cellular level, now we will be graduating to the neural intelligence level. We have done the genetic intelligence, we have done the intelligence of a cell, we have done the intelligence of energetics, that is mitochondria. Now we will be talking of neural intelligence. Watch out for the next talk. Until then, bye bye, good luck, see you.